Hi, my name is Ziynet Nesibe Kesimoglu. I'm a PhD student at Marquette University under Dr. Serdar Bolda's supervision. Today, I would like to present my poster about inferring competing indigenous RNA interactions in cancer. ceRNA stands for competing indigenous RNA afterwards. The underlying biological processes are governed by multilayered regulatory networks, and dysregulation in these networks causes diseases such as cancer. Recently, an regulatory layer related to microRNAs has been discovered, where certain RNAs targeted by common microRNAs compete for these microRNAs and thereby regulate each other indirectly by making the other RNAs free from regulation. Such indirect interactions are called ceRNA interactions, with their essential roles in cancer. Inferring ceRNA interactions experimentally is cost and labor prohibitive. Therefore, several computational approaches have been introduced. Despite efforts in this new area, there exist crucial drawbacks in current computational approaches. Most studies start with differentially expressed mRNAs. However, the inclusion of all genes including the non-coding ones is important. Current tools compute only pairwise CRNA interactions. However, inferring groupwise CRNA interactions should be considered. Expression is assumed as unlimited in the cells without considering the expression of microRNA and target is sufficient or not. Furthermore, two genes with many common CRNA partners might be inferred even they do not have direct CRNA interaction. So excluding these indirect interactions is important. In this study, we developed a computational tool named CRINET to infer cancer-associated CRNA interactions and groups genome-wide to address the aforementioned drawbacks. I included the links including GitHub link for the code and blog for the tool. Also, this study could be found at bioRxiv. The first figure is the CRINET workflow. The first step is data preparation. The next step is computing microRNA target interactions. In the third step, CRNA interactions are inferred and the preliminary CRNA network is generated. On this network, the indirect CRNA interactions are excluded and the final CRNA network is generated. Finally, ceRNA groups are inferred and adapted to the ceRNA network. We applied CRINET to more than 1100 breast tumor samples from the cancer genome atlas and inferred 17,443 CRNA interactions and 81 ceRNA groups. In our workflow, we computed microRNA target interactions that we obtained from target scan, integrating with expression datasets. Moreover, we introduced interaction regulation score to consider the sufficiency of abundance for microRNA target interactions, and also introduced an effective regulation score to exclude genes that are not under the strong regulation. While inferring ceRNA interactions among RNAs, we consider significant overlap for common microRNAs, some collective regulation metrics, and we excluded the indirect interactions inferred just because of the amplifying effect of common CRNA partners. After applying a community detection algorithm named WalkTrap, we inferred ceRNA groups and adapted them into the network. Since we built the ceRNA interactions relying on microRNA target interactions, proper selection of these microRNA target interactions is important. Therefore, we evaluated each filtering step of microRNA target interactions with protein expression benchmarks, and we had increasing performance on each filtering step. As in the Qubit paper, we evaluated ASR1 protein deeply to show the anticorrelation among microRNA expression and protein expression. In Figure 3, we showed the heat map of protein expression for top and bottom 10% samples that are ranked with respect to microRNA expression and clearly showed the discrimination between two sample sets. Also, we measured fold changes for these sample sets and had consistently good results for CRINET, while qubit inferred regulators had some low fold changes. We also checked the CRINET eliminated regulators, which does not show any pattern and mostly having low fold changes. To assess the accuracy of ceRNA network, we used Link's database knockdown experiment and inferred ceRNAs were able to predict gene expression change of their CRNA partners with high accuracy, while different runs with Hermes was not as good as CRINET while it was running with the same dataset. Links database, knockdown experiment, and Inferior were able to predict gene expression change of their ceRNA partners with high accuracy, while different runs with Hermes was not as good as CRINET. Moreover, Inferior and ceRNA groups were highly enriched in cancer-related processes and non-cancer genes with significant p-values. When we check the Gaussian terms for Inferior high-degree genes in the inferred network and some inferred groups were consistently enriched in immune response-related processes. In this study, we developed a computational tool named CRINET to infer cancer-associated ceRNA interactions and groups genome-wide, addressing crucial drawbacks. We consider sufficient abundance, we consider effective and collective regulations, and we excluded indirect ceRNA pairs. When we evaluated our results, inferred ceRNAs and groups were significantly associated with known cancer genes and processes, suggesting their important roles in cancer. Inferred ceRNAs and some ceRNA groups and high-degree genes were consistently enriched with immune system-related processes, suggesting that ceRNA regulation could have an important role in immune response in cancer in the light of the studies confirming that the weaknesses in the immune system function is closely correlated to the tumorigenesis. In conclusion, getting the benefit of multiple biological datasets, this approach facilitates a better understanding of ceRNA regulatory mechanisms, solving important drawbacks, which would shed light on the underlying complex regulatory circuitry in disease conditions. Finally, I would like to thank my advisor Dr. Serdar Bozdağ for his guidance. I would also like to acknowledge the support of the NIH for funding this study. Thank you for your attention.